that we have in our curriculum good instructional techniques. The students are learning the techniques that they need to learn, but they don't have this aspect of doing real research. So we're hoping to keep the instructional positives that we have and then add this other aspect.
Well, what we were trying to do was to give our students an opportunity to have the experience of doing authentic research. We have a very large number of students in the department. Very few of them get to do research individually with faculty members, so we wanted to give more students that experience.
So the idea is that even in bacteria like E. coli that have been studied for decades, there still is quite a bit we don't know. And one of the things we don't know is how many very, very small proteins are being made or encoded in the genome, and if they're being made, what they do. And since this is really a black box, it's an ideal project to give to undergraduates, to present it in a way that they choose a potential small protein to test to see if it's really being synthesized, and then if it is, give them the opportunity to try to characterize it in the future. And to date, the students have identified 16 new small proteins. And they're being characterized by those students in the lab right now. And I think they've got some really interesting proteins they've identified, and it's going to be exciting to see what they discover.
My greatest expectation was that the students would develop a greater interest in research, but also you're interested in techniques and giving the students the tools that they're going to need to go on to graduate school and compete for jobs.
So we found that it's been very positive. The student response has been positive. We've been just started the formal assessment, but we've been getting class evaluations for a number of semesters, and 90% of the comments that we're getting from students are positive, and they're kind of the comments that you'd like to see, such as I never considered doing research, but I'm really interested in it now, and I'm thinking about doing independent research in the future. I think that what Larry and I have both found is that adding authentic research to a real standard school curriculum or college curriculum is challenging, and it's time consuming. It's also very rewarding, but there are extra aspects that require a good bit more effort on our part outside of the classroom.
Because of that, it works best if the research that you're introducing into the laboratory is research that you yourself are doing. Mm-hmm. So you are already investing in that project, and in fact, the results of the students will further your own scholarly activities. But also, I think for both of us, we want to help other faculty members in our department and other departments use this model, where it's already starting to be done. Our genetics class is starting to use it, and they're designing a new cell biology laboratory class which is going to use this general idea. And at the introductory level, I'd like to explore engaging additional faculty in that specific class to do other projects.